Most pleasure to be with you this morning. I thank Dr. Hadi and Dr. Muhammad for this kind invitation. We are neighbors, but nevertheless, we meet in conferences, and I look forward to a bright future where we can collaborate uh, closely on a scientific basis to generate scientific data and therapeutic modalities to the rest of the world. My name is Khaled Balto. I graduated from King Abdulaziz University in Jeddah, the west of Saudi Arabia. And then I got my certificate in endodontics and doctor of medical science from Harvard University. By the way, I was accepted at Loma Linda with Dr. Mahmoud Tourabinjad, my dear
Oh,
friend.
really?
I was so lucky. But then things just switched toward Boston. But nevertheless, Dr. Mahmoud has visited me in my home several times, him and his wife, and I continue to hold a very strong brotherhood relation with him. I stayed in Harvard, Boston for about 5 years, where my endodontic research was focused on modulation of periapical bone destruction response by manipulating some cytokines. I specifically worked on interleukin 6 and interferon gamma. Our work has been recognized at that time. It was the year of 2000. 2000 might sound so far away to the young residents attending, but for me, it sounds as if it was 2 days ago, where it merited the first research in the United States of America that year. And our work has been published in Journal of Immunology and Infection and Immunity and so on. At the moment, I'm a professor at the University of King Abdulaziz University, and I was among the collaborators who started the Saudi board programs. We started the Master of Science in Endodontics programs in the university, and at the moment, I'm finishing up the PhD program. Nevertheless, I've been working with the companies across the world as opinion leader in R&D, and that's where I met the elite people like Dr. Hadi and the colleagues from Iran. So this is a glimpse, a short glimpse about me, ladies and gentlemen.
Thank you very much. It's a very great honor for us to have you here, and we're all ears to uh, carefully see and hear your presentation.
Thanks, Doctor. So in this virtual room, do we have residents with us?
Yes, yes, we have our residents here. Uh, they're looking at you, and we have a class actually, and you are now the presenter of our class.
Okay, great. So if anybody has a question, I think anybody can stop me at any time. I really look forward to for this discussion. To see how people think about different things, and let's go. All right, ladies and gentlemen. So the title of the presentation I have chosen when Dr. Hadi approached me was to the paradigm shift in endodontic microbiology. I know some of you might ask, yeah, Dr. Balto, why you have chosen to talk about microbiology per se. It's very boring subjects for lots of my residents here. However, I have chosen this topic because of several reasons. You know, ladies and gentlemen, that recently endodontics has changed tremendously. Apart from the technological advancement, microscopes, calcium silicate materials, but our understanding of the bacteria host interaction is tremendous. Uh, if you allow me to go back just one step backward and review with you what is the aim of root canal treatment. Look at me and you. We every day we woke up in the morning and we walk to our offices or to our clinics and we do root canal. Why we do that? You know, the question might sound philosophical, but if you put it within the language of words, I think we can use two words. We do root canal treatment to treat, and we do it to prevent. Okay, treat and prevent. Of what? Of infection. Infection of what? Of a disease called periapical periodontitis. Lately, we have understood the role of dentin as a bioactive substance. Dentin has been recognized traditionally when I was a student, pre-grad student, that it's enamel and then dentin and then pulp, and then from outside is cementum and bone. But those physical barriers, what appeared as physical barriers, are not only physical barriers, they are bioactive substance. We have learned that just a few years ago. That's why when I talk about treatment and prevention, I have to add and or pulpitis to the apical periodontitis. I tell you, apart from my work at the university here, I work in private practice 2 or 3 nights a week. And a big part of my treatment now is regenerative endodontics. And, you know, for me, it was easy to understand and to teach that not only to our residents, even to our interns, that, you know, vital pulp therapy is something that we got to think about tremendously, because I believe, like Walters did a couple of years ago, that there are lots of teeth that receive root canal that they don't need or they didn't need to have a root canal at the beginning. So this is the big picture, you know, that we do treat and prevent of pulpitis and apical periodontitis. But, you know, we still have one trick here. Is apical periodontitis really a disease? Well, believe it or not, in my career, I have asked this question several times, and you're going to be surprised to know that most of the response that I got, no, periapical or apical periodontitis is not a disease. It's just a variation of dental caries. It's just like a trauma. You know, you were hit with a trauma. If the trauma is a disease, trauma is not a disease, but trauma can cause a disease. But if you consider the definition of a disease, you know, it's a pattern of response. You see, look to the words, pattern of response to some form of insult or injury resulting in disturbed function or structure. So this disturbed function could be organic or could be psychological. So the trauma that results in psychological disturbance, that's a disease. So in the essence of definition of a disease, apical periodontitis and pulpitis is a disease. And every time I look to the X-ray or I look to a CBCT or I listen to a patient's chief complaint, I have to determine the disease. So like yesterday, I was with the residents in their clinic, and this lady has walked in with a history of a disease, a pain in the upper first quadrant. Radiograph shows normal looking teeth. Except the history, there is nothing else was significant. And yet the student wanted to treat or do RCT to tooth number 16. So I asked the student, what is the disease you're trying to treat in here? She had pain. All right, she had pain. It could be periodontal. It could be trauma from occlusion. It could be TMJ. There is signs of wear facets. It might be a Bruxer. Maybe the pain has been there. So if you keep this in the back of your mind, student, you know, every time you look or you're gonna start a treatment, you have to recognize the disease, because there must be a performance indicator that the signs and or symptoms of this disease has vanished upon successful treatment of yours. All right. So if we agree on this principle, ladies and gentlemen, I want to ask a question. I wish you were in front of me here. what is the etiology of apical periodontitis as a disease? Well, obviously, we cannot have interaction here, but what is the etiology of apical periodontitis? I know you might say, what is this silly question? You know, it's not silly because before we know what we know now, people in the hundreds and maybe hundreds of years ago did not know. They thought that there is worm go inside the tooth and cause this disease. Until a couple hundred years ago, the people thought that, you know, when the necrotic tissue become in the pulp can cause apical periodontitis. So the knowledge that we know now did not come from ease. We had a progression in our understanding of the etiology. Of course, you know Kakahashi. Do you? This is my favorite article. You know, I always use it with my pre-grad, post-grad. Actually, a couple of years ago, I was invited to Harvard University to be external examiner for their master program. And my main question was about Kakahashi, believe it or not. It was very interesting because the Kakahashi article, as a principle, everybody knows it, probably. But it had lots of details that very little people, you know, thought about it. We'll talk about in glimpse. But you see, this Kakahashi article, though it's a very low technological article, it was not PCR based or molecular assays or anything. It was very, very simple, you know, animal study that did not incorporate any high level of technology. With that came the other studies of Sangwist, and I'm sure you residents, Indo residents, maybe know these names. I'm very hard on my student when it comes to endodontic literature. Sometimes we have mock exams about the authors, Toro Binjad, whom where you can cite Mr. Dr. Muhammad Toro Binjad, and how about Hargraves? How about Kakahashi? How about so these names are very important because that's the heritage of our specialty. So Kakahashi, Sangvist, and Mueller are landmark studies that has taken us close, closely to understanding the real etiology of periapical periodontitis, where the gist or the conclusion was microbiological etiology that is bacteria. So it looks like that all of us know bacteria, but you know, bacteria is one element of the microbiology. There are lots of other elements, and I always thought, why is always bacteria? We know, you know, the emerging diseases we have right now in the humans, we've never witnessed it before. What causes Alzheimer? Have you thought about it? What causes methania gravis? And there are lots of diseases that bacteria is not standing there. Now we started to hear about non-microbiological etiologies. We started to hear about viruses and what virus can do, and we learned great lesson during the COVID pandemic. Who could imagine that viruses can cause blood clot? I was not taught that. And even the scientists, when we were living in the pandemic, when they understood that the blood clots that were killing most of the people who had COVID was coming from viruses or collateral effect of viruses, we were astonished. So what other things viruses can do pathologically that we didn't know? So bacteria is a big cover, but yet let me go to the bacteria first, ladies and gentlemen, and then I'll pull out and try to shed some thoughts on the other microorganism. Okay, this image, I'm taking it from Kakahashi in 1965. It looks so beautiful. Sometimes I sit in my office. This today, I'm sitting in my office in my home. You know, I turn off the light and I look at this picture here. Wow, this is a picture of a rat molar. And this Mr. Kakahashi did a pulp exposure in these teeth, and he left the teeth open to the ORE environment. He let the rats live for a couple of weeks, 3 weeks, and then rabbits, not the rabbits, sorry, the rats were sacrificed. And look at the pictures. All of these black thing is the PMNs, probably severe periapical bone destruction. The inside the canals looks empty, and so on. But Kakahaji shows another big difference image where the germ-free rats that received pulp exposure and lift open, the same situation. By the way, all of these rats were male. I'm not saying that Kakahaji was racist toward male rat. No, he chose uh, males to standardize. You know, with the female rats, the problem with estrogen can cause bone destruction. It could be a masking effect. So they were males, the same age. They were raised up in the same lab. They have the same water. They have the same filtered air, and the only variable was that this group of rats were germ-free. It's a very special brand of rats present in Jackson laboratories in upstate in Maine. And this group of rats were uh, usual one rats that they have commensal bacteria and so on. Look to the contrast. The germ-free did not have any bone destruction. And even inside the root canal, the pulp, converted to fibrous type tissue. So there was no disease, basically, in the germ-free rats. Taking it together, the only difference between the two groups were the germs. But somehow Kakahashi and his colleagues choose bacteria. So Kakahashi did not do any microbiological studies to know exactly in the conventional group of rats were they bacteria and other maybe microbiological agents or only bacteria. But ladies and gentlemen, this is not the only thing that is cool about this study. Look at this image. In some of the rats, and most of them, there were bridging of the exposure site by dentin. No calcium silicate, no control of bleeding. It just happened. This is, for me, is really significant because with the periapical bone destruction, the microbes were different. And with this image that you see in front of you, it says to me that the biologic system is capable of healing and regenerating if there is no contamination, microbial contamination. Isn't that what we're doing nowadays with vital pulp therapies? We're giving a chance to the pulp to heal. We were told that pulp can't heal. You remember the Van Hassel higher studies, Chem, pulp is low compliance system. Once inflammation starts, it will never stop because the pulp will strangulate. It will not withstand edema. But this was not true. We know now pulp can actually withstand the pulpal inflammation tremendously. And they do harbor or recruit stem cells that bridge off or initiate or make significant healing with new tissues. All right. So the progression that we had so far, we argued about the periapical periodontitis as a disease, if it's a disease or not. It is a disease. What causes a disease? We talked about the classical studies of Kakahashi. And by the way, the other studies of Moeller and Sangvist, these just augmented the finding of Kakahashi in other models, Moeller in monkeys and so on. So we have this piece of knowledge beyond any doubt that this is what is causing apical periodontitis. But I alluded to you a little bit in the last two slides that Kakahashi and even Moeller and others did not do any bacterial or microbiological investigation about the nature of the microbes. So I want to ask you, what is the detection methods that we know for microbes? Of course, everybody is going to say number one, culture. And that's the history of recognizing bacteria. We cannot see bacteria by our naked eye, nor can we see viruses, nor we can't see fungi, not even archaea, not even prions, the super proteins, and so on. But we take what a sample of whatever we think it has bacteria, we culture them, we provide the food, the nutrients that is necessary, and we look under a microscope and we characterize them. But nowadays we have other detection methods, and the most important disadvantage of the culturing method that you cannot culture something you don't know unless by chance it comes up. But you cannot put a culture and you say, "Oh my God, I found a new bacteria." It will never happen because we don't know this new bacteria, what nutrients they know they need, what redox potential they need, what oxygen maybe they have a special nutrients. We will not know until we isolate specific bacteria. We know exactly the nature of their microbiological interaction and so on. So that's why cultures remain limited. If people have a urinary tract infection, we have to do culture for them to know what kind of bacteria are there so we can give the appropriate antibiotic. But we didn't do that in endodontics. We didn't take a culture from the pulp when you open it, or sorry, a sample of bacteria or microbes, and then we culture it to know what kind of antibiotic I'm going to give, clindamycin or augmentin or whatever, cephalosporin. We don't do that because basically, the disease of the periodontitis is polymicrobial, not monomicrobial. What other methods? Immunological methods. That in the immunological method, again, you're gonna detect bacteria that you know because you have a specific antibody through which it has a tail that is clocked to its antigen, and you can quantify that. But we have advanced more with the molecular genetic technique, PCR, derivative, RT-PCR, and the difference between PCR and RT-PCR are very important, students, because with the PCR, we're dealing with DNA, and I can't detect DNA from a cell that has died because DNA does not vanish right away after bacteria is dead. However, when we take the messenger RNA, if there is a messenger RNA, it means there is protein synthesis. And protein synthesis cannot happen unless it's coming from a live bacteria. So that's why RT-PCR or the messenger RNA might be more accurate of the live bacteria interaction. The classical studies that we had before from endodontic surgeries, they take biopsy and they analyze it, or histologically and microbiologically, and the hit on those studies were that maybe the DNA were taken from dead tissue already, and it doesn't make sense that bacteria can go outside in the periapical area and live great life. And what the immune system is doing if bacteria can persist in the periapical granuloma? Where is the recognition system of the macrophages and other things? So this kind of debate is always present amongst microbiologists and immunologists that, you cannot tell me, like Walton said before, a couple of years ago, that bacteria can live outside the root canal in the periapical area happily. Well, you have to convince me then that there's an immune tolerance that happened at that site of the periapical lesion that mask off the recognition system of our body to recognize there is live bacteria inside. This is a big controversy. We can talk about another time, but nevertheless, I wanted to open some glimpses in your brains about thinking critically on data that you have read or you're reading. Is everything okay here? Is there any question, Dr. Hadi, or shall I continue on?
I don't think so. I don't think so. Sorry.
Everything is good? Carry on?
Yes, yes, please go ahead.
All right. And the fourth thing is ATP based. Again, ATP will come only from live bacteria. So the detection method have enriched so much to know the type of bacteria. You know, the DNA checkerboard that has been developed by Sokransky in Boston, actually from Forsyth Institute. That was amazing. It will show you hundreds of bacteria as signals here and there, and you can maybe detect a band that you didn't know, and you go further to make a probe and so on. So microbiology has witnessed paradigm just in the quantity, quantification, and in the identification directive. Okay, the interesting question that I was starting my discussion with from the beginning. Bacteria as an etiology of periapical periodontitis, we know it a lot. And I trick my students sometimes, especially level one, they're still young and they don't understand a lot of endodontics. What is the etiology of apical periodontitis? And you find the answers to the answer, Fusobacterium nucleatum, Enterococcus faecalis, Teropomia denticalis, and so on, blah, 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 blah. Well, it's not a mono infection. Otherwise, we could treat periapical periodontitis by antibiotics or other immune-mediated agents. It's polymicrobial. Of course, it's not, I'm not saying that all of these bacteria in front of you is going to be present in a sample or biopsy you take from the periapical lesion. I'm saying all of these bacteria have been identified by several investigators, and some of these bacteria don't like each other. If they're present, they're going to kill other bacteria. Some of these bacteria love each other. If you have one bacteria like Troponemia denticalis, maybe you'll find Fusobacterium by an odds ratio close to 5, and so on. So the interaction between bacteria is another significant word, especially when you take biofilm into consideration and the communities of biofilm. So if you close your eye once and try to imagine this biofilm, there's water go inside them. The pH is very acidic. You have like people like Gabila, of Troponemia living there. They hate the other niche there. It's a whole different world. It's amazing. Um, and I was telling my colleagues once when I was giving this lecture, I used to work in the Forsyth Institute in Boston. Forsyth is affiliated with Harvard University, and it's just, a short walk from 188 Longwood Avenue to Forsyth. There was this famous microbiologist. His name was Sigmund Sekranowski. He's the one who invented the DNA checkerboard. Let me tell you this story. It's very, very interesting. And Dr. Sokransky is a very, he was a senior citizen at that time, but he was a very well-known microbiologist from dentistry. Most of his studies were in periodontal pathology. His lab was next to my lab. So every time I go into the lab with Sokransky to use his oven, I find him sitting in front of the oven of the bacteria, and he was sitting like this, looking to the oven. So, I'll go back and forth, maybe 1 hour or two, and he's still sitting there. Maybe he will have a cup of coffee, and he was sitting looking like this. So one day I asked him, Dr. Sakanis, what are you looking for? I mean, I see you every day or every other day. You're sitting in front of this oven of bacteria, and are you imagining things? He said, no, no. They can't pronounce my name, Khaled. They say Khaled, Khaled. Khaled, I think bacteria are very interesting organisms. They could be like human like us, but in a very small scale of dimension. And when I sit like this and look at them, I imagine how their life would look like, because they have life. I said, "Oh, okay, Dr. Sumik Shukran." I didn't understand what he was saying. Actually, it took me like almost 15 years to understand. Oh my God, that guy was ahead of me by 15 years already when he was trying to understand the how bacteria interact with each other and how enormous this dimension of life is that we can't appreciate because our eyes cannot see it. But I took what Sigmund Sikorsky told me and taught me, and I realized that there are viruses and there are archaea. You know what's archaea? Archaea is very sophisticated microbes that we don't talk about. I don't know why. Though I think, I might be wrong 99.9%, that most of the diseases that are not treatable nowadays can be caused by archaea. And we have fungi. All of this together makes the complex of microbes that could act as synergistic or antagonistic to the periapical bone destruction. Now, most of the studies, ladies and gentlemen, in the microbiology of root canal from inside the root canal or outside, they found most of these bacteria is number one, and there is viruses. But not everybody has been looking to viruses because they say it's not significant. Even if I find Epstein viruses, what the hell Epstein ABV will be doing in the periapical lesion or in the necrotic pulps? Or the study that found fungi and archaea. So these microorganisms are there. Why we have not investigated them? Does this justify some of the refractory cases of apical periodontitis that not work? Does that make us speculative that our antimicrobial ways of in clinical endodontics might not be enough, and we have to go to a higher level? Well, let's quickly go over them one by one. All right, viruses. I told you, I always thought viruses means influenza. Viruses mean AIDS. But when I dig deep in it, not deep, I started to read about it. I learned a big lesson from Corona, as I told you. We're talking about 1958 and Roche and Schindel in 1960, very, very far away. And those people did very preliminary, basic studies. And look, these data have been there since 1960. That apical periodontitis has a strong association of the presence of Epstein-Barr viruses uh, as much as 72% or 75, 73%. Not only that, it has been concluded from the series of studies that have been carried on the Epstein-Barr viruses that the more the lesion size of the periapical periodontitis equal or more than 5 millimeter, the more chance that the Epstein-Barr virus is going to be there. We're talking about 91% person. 19 cases out of 21. This is significant. Don't tell me this is by chance. Don't tell me that the lesion got bigger, so the immunity went down, and from where the viruses came to bring with to begin with, and how come the viruses
Excuse
we
me,
know.
I have a question. Can you please provide us with the title of this article? Because I searched a lot about the primary or the principal study talking about the etiological etiological influence of viruses in apical periodontitis, but I couldn't find anything other than a case report which was written by Slots. I think it was 1980s or 90s or something. Can you please provide me with the title?
You mean the study of Rauch
Yeah, yeah.
and
It's the
Schindler?
current study
Okay,
that
by
you
all
mentioned.
means, I'll send it to you by email
Oh, thank you.
or you see, I'm not sure I have the original article or it was cited in this in Handia in Journal of Endodontics in 2010. Whether both of them, I'll send it to you.
Thank you.
But there, I think I have a subfolder that I call it viruses. It has several studies. I'll share it with you on Google.
Thank you.
All right, you're welcome. So, going back to the point, my mind is just a spectacle. Okay, I'm reading the data and I'm trying to make stories out of it. So maybe one scenario we can look at. I'm not talking about uh, Rush or Schindler per se, about the bulk of the studies that we have. So, if this is the case, why wouldn't you use antivirus in our refractory apical periodontitis cases? Oh, well, why you are laughing at me? If anybody of you is laughing at me, look to the data. It says viruses are there. So what the viruses are doing, we don't know yet. We're still speculative about it. But there are viruses in these refractory cases. Okay, how viruses destroy. I put three here or four bullets, but I'm very cautious about it because I understand my position at the moment after the pandemic, COVID pandemic, we are yet to understand this creature. Actually, if you want to be philosophical, and I like to be philosophical sometimes, are viruses creature? Are they? They don't have face. They don't have not face. They don't have
Big
picture.
question.
Oh, we have picture. Viruses, viruses is just nucleic acid. So I don't know. You see, it can be so complicated. Well, I'm not. I don't know what viruses are honestly, but it's something that causes a big array of things when they go inside biologic system. So the theories that I honestly I got it out of some articles and internet data that the first method of destruction is direct cytopathic effect on the infected cells. It invades the cells, it screw up its molecular genetics, and it makes it make its own protein and so on. Okay, we understand that, and that's how the influenza virus, for example, works. But there are other two mechanisms that I found, which is generation of oxidative stress and whatever, B lymphocyte or others, but the net result of it is the activation of Rankel pathway and OPG. That causes bone destruction. It's pro-inflammatory and inflammatory pathways. It recruits osteoclast, activate osteoclast formation, and then you lose mass of bones. When oxidative stress destruction happens, I don't know. Honestly, I'm just again what I'm trying to do here is to provoke your thinking abilities, ladies and gentlemen, so you can be critical appraisal. And last but not least is the production of TNF alpha by again B cells, which is a major mediator of tissue destruction. So basically, it's the cytopathic effect or indirectly through activation of Rankel pathways or TNF alpha, which is inflammatory cytokines that you know. Okay, so viruses can cause bone destruction indirectly, according to these data I'm showing you. Okay, then why we are only using irrigations that only disrupt bacteria and protect tissues, and why don't we do higher level research to see exactly what these viruses are doing inside the root canals? Taking it all together, that I can share this with you, Dr. Hadi. Maybe we can do research together. Viruses do not usually cause periapical infection by themselves. At least that's my gut feeling. I don't have any evidence on this. Can they initiate? I don't know. But at the moment, most of the people around the world think that viruses may contribute to the severity, persistence, and immunomodulation of the lesion. This sounds much better to our knowledge. Yeah. So if there is an exacerbation or complication of a lesion, we think about atypical infection. Comes number one of them is R viruses. So with talking about this dimension of microbial interaction in the root canal system and or periapical area, we should start thinking about viruses. I'm not saying go crazy and start antiviruses. Maybe, we think about research. We cannot use humans as guinea pigs and start giving drugs on them while we're not sure. But very little studies of any, I've seen that investigate this directive.
May I ask you a question here?
Yes, please.
Uh, you definitely know that in large amounts of samples, we have necrotic tissue or even nothing within the root canals infected uh, after an open necrosis or after an unsuccessful root canal treatment. So, in these cases, is it possible for a virus to survive to cause any kind of infection to in order to have a kind of persistence or modulation or to increase severity of infection over there? Is it possible for a virus to survive? As far as I know, any kind of virus needs a vital tissue to invade, a viable cell to invade and to proliferate. So in case we do not have any living tissue over there, so what can harbor this ecological factor?
That's a very, very good question. You see, um, just a few minutes ago, I speculated if viruses are really creatures, but
Yeah.
well, we have two kind of necrosis: coagulative necrosis and liquefaction. For viruses, our understanding at the moment, for viruses to persist and live, they need viable cells. This is the only way they can live. Though, Dr. Hadi, there are lots of studies that showed that DNA has a very high affinity to dentin surface. And even some of the studies that looked into the DNA, from the dentin chips, they said this DNA might have been released from cells that died just before we collected the sample, and the DNA adsorbed to the dentin surfaces for a while before it is destroyed. The gold question is here, Dr. Hadi, is what is the half-life of DNA when it's outside a cell before it gets destroyed and digested? I don't know. It depends on the, as you said in your statement about the environment, how acidic it is, how basic it is there compounding factors and so on. So, anyways, that's a good question, but the answer is not present at the moment. All of these data that I shared with you, ladies and gentlemen, is the data of our colleagues across the world, and they have isolated the viruses. But that's a very good question to investigate.
You know, just some research point came to my mind a year ago to have a kind of intracanal medicament to be a combination of, for example, calcium hydroxide and an antiviral agent to be placed inside the root canal and to see the effect of this kind of medicament on patients' pain or, healing of periapical lesion. But the point was that in case we do not have any living tissue over there, so there is no virus. So there is no need to add any kind of antivirus agent to the medicament. So it's a useless idea that way.
No,
In
no,
case we probably inject such a thing inside a periapital lesion, it may make sense. But inside the root canal, which is devoid of any living tissue, I think, this raised a big question in my mind.
you see, this is how genius question starts, Dr. Hadi. I agree with you 100%. But I would like to attract your attention why you are hesitant to accept that there is viruses inside the root canal. Because you see, in the disease root canal, like the cases that we opened yesterday in the student clinic, some of them were hyperemic. Some of them were irreversible pulpitis, but there's some blood, so there's cells inside. Okay, if you are talking about the extreme progression of the disease toward necrosis, okay, there is liquefaction necrosis, and there are cells in the liquefaction necrosis, there's PMNs. I don't know if viruses can go inside humans or not. If you're talking about the very, very, very extreme liquefactive necrosis where it's dry necrosis, I don't know. How about if there's some cells stuck inside lateral canals, that were not destroyed, and viruses are using that as machinery base and getting stuff outside. I mean, that also thing can happen there. But you see, we have to build on the results of the studies that we have in hand already. All of these, I mean, if I and you come one day, okay, let's get samples from 100 cases randomly that I'm going to open this week. We're going to isolate viruses. So the fact that viruses are present inside disease root canal is beyond any doubt. But then now we want to investigate more about what is going on, what governs them. Are they persisting? Are they fading away? Are they modulating, like this slide in front of you is saying? Okay, if they're modulating, how come they're, do those cells finish? Whoever the remnant cells in the lateral canals or an isthmus, maybe. So we still need a 3D thinking pattern to try to understand what's going on with viruses. You see, a virus is very interesting. Uh, I don't know if you would like to call it a creature. I would rather call it phenomenon, viral phenomenon. Okay, how about archaea? Students, what's archaea? You're lucky they're not listening, sitting in front of me, because when I give lecture, I always, get everybody involved. So to get everybody involved and awake and don't fall to sleep. So basically, look at this diagram. We as human, are we eukaryote or prokaryote ourselves? Are they eukaryote or prokaryote? Who can tell me? I asked this question to one of a class a couple of years ago, and the guy who was sitting right in front of me said, "We are prokaryote." Prokaryote. I said, "Maybe you are prokaryote. I am eukaryote." So eukaryote is they have nucleus, as you see here, and the DNA inside the nucleus. Prokaryote, the DNA materials in the cytoplasm. So we, the eukaryote, are more organized. But archaea are actually eukaryote. They are very civilized kind of organism, microorganism. I cannot say they are bacteria because they are not bacteria. Why they are not bacteria? If you don't know, quickly, archaea are microorganism classified as the third separate domain of life. They are a big issue, and very little of us talk about them. I don't know why. They are separate domain of life. Do you read that? What's what is the problem here with these organisms? Well, number one, archaea are present inside endodontic root canal disease, disease root canal. 85% of the sample that has been PCR amplification have found the presence of archaea. And the incidence or the presence or absence of these microorganisms have varied according to the authors in different settings. But like viruses, archaea are there. So if you look here, this is a very interesting study, Dr. Hadi. You see, his name is Birzkis. I think he comes from North Europe. I'm not so sure. In 2018, all of these arrays. So at least these people have cared to use the molecular techniques to see if there is archaea or not. They were archaea, amongst other, to Tanarella forsythis, to Rubemia dentacalis, blah, blah, blah. The whole gangs are there. With them were archaea. And look, it's closing to 50%. 50%. 50%. That's a lot. All right. And when they compare the primary endodontic with the post-treatment disease, like failure, the archaea has been always found in the primary endodontic diseases more than secondary later post-treatment disease. But there's variation. I don't think it reached to the level of significance. So, which means they are present in both primary and post-treatment disease, maybe with lower presence in the post-treatment disease, but does not escalate to be significant. So it can be hypothesized that archaea may participate in endodontic microbial community. This is goes without any doubt. That's why I included it here amongst the other. Now, with that being said about archaea, ladies and gentlemen, the rest of the analysis of this third life of domains goes on, and it was loss. It's gonna be lots of microbiological talks that probably not going to be interesting to clinical endodontics. I just wanted to attract your attention about archaea, and maybe you want to read about more, or maybe you can gather together one day and talk about it critically. Okay, time is going. Let's talk about fungi. Uh, Fungi are present maybe in lesser amount or percentage reported in reported cases, but fungi are known or characterized by producing hydrolytic enzymes that may be involved in apical and periradicular tissues. And recently, other types of fungi have been determined to be associated with the presence of type of fungi and other cells or bacteria. Now, when I was a student, we were taught that we should not use amalgam in retrograde fillings that contains zinc. At that time, we used to do periapical surgeries with amalgam. That's a history of endo. But why with zinc? Why zinc was bad to be in amalgam? It had to be zinc free because zinc is a very rich media for growth of fungi. And there has been reported cases, it's reported already at that time, but the reports are still there, that people have been diagnosed with aspergillus infection, which is kind of fungi, because of the zinc content. And aspergillosis is very stubborn type of bone destruction. People can lose half of their maxilla, if it's upper molar, but because of the aspergillus infection of the fungi. So fungi do have a significant method or mechanism by which they can destroy the bone in general. So these are the type of fungi that has been uh, isolated in the periapical area. Aspergillosis comes as number one. I don't know about the other, but Candida albicans has been isolated in several studies before. But for me, there are two types of them that stands out: aspergillus and the Candida albicans. So, what is the interaction now between microbiome and bacteriome? That's very interesting subjects that is very rich in discussion in the microbiological world. When you go into chat box with the microbiologist, this is a very important thing because, like dentists, we were talking last month. We had journal club at Harvard University for the alumni, and we were talking about cervical root resorption. You know, and for me, I said, guys, cervical root resorption does not its successful treatment does not depend on me as endodontist. It's multidisciplinary. It's me and the periodontist and the periodontist. I'm not going to be a Superman to treat the cervical root resorption. It's not within the scope of my specialty. So The big thing in dentistry now is the multidisciplinary approach of treating several diseases and reconstructing several anomalies in the head and neck region. The same thing is going in the microbiology world. It's now the biome interaction. It's in their mind, it's not bacteria alone, it's not fungal alone, it's not archaea alone, it's not virus alone. We have to have like multidisciplinary approach. This is like multi-biome approach. And lots of studies nowadays, Dr. Hadi, on these is biome based. Look at the amount of these studies I found about microbiome and bacteriobiome in, in general and in the dentistry a little, that these two biomes are independent but often interact with each other. When they interact, they result in treatment resistant of microbial community. That's very significant because when we see lots of cases that root canal looks perfect in radiographic and CBCT. The lesion persists. You go ahead and go do a surgery, make sure there's no crack, there's no other route for bacteria to go outside, causing the persistence of the lesion. You go and do surgery, you take your biopsy. If I'm lucky, I can do maybe microbiological analysis. And most of these reports come that there is viruses and there is fungi and there is bacteria, you know. So, okay, maybe that resistant disease could have faded or shrunk a bit with antiviral medication, maybe systemic or maybe injection or whatever. That's something that maybe we have to gear up for a couple of years from now, where treatment of endodontic diseases can be the same as we do or escalate to be kind of medicine, like internal medicine. We give medication with some surgical, non-surgical treatment. This is how we can imagine things can be. For example, I was asked in an interview last month or 2 months ago, what would be the future of endodontics? I said, honestly speaking, I think in a couple of years, maybe 50 years, I don't know, there will not be endodontics because the stem cell research is going to boom, and we're not gonna make a treatment that is resulting in dead space inside people mouth, and we say that we did them a favor. It's not really healthy to have a dead space inside a human being because you can't eradicate bacteria. You reduce infection, disinfect, but you can't sterilize. So I think the best root canal filling material that is going to prevail in a couple of 100 years or maybe tens of years will be the blood, not the gutta percha, the blood. So we're gonna fill root canal with blood, pretty much like we're doing nowadays. Don't you guys do revascularization? I do it because if I can do it and use Revask, that's a better root canal filling for me than gutta percha or bioceramic or whatever it is. All right, so please keep in mind this microbiome and bacteriome interaction, the multidisciplinary, approach for microorganism that causes diseases in our body. Okay, we have to go a little bit faster. All right, so the ecological factors that are important for bacterial interaction, there are receptors, there's nutrients. Nutrients is the source of food. What is the source of food of bacteria inside the root canal? Well, no, everybody's gonna say, coronal leakage. Okay, of course, coronal leakage. But how about, stuff that you leave inside the root? A canine that is very wide, labio palately. You leave untouched surfaces that harbors some tissues. This tissue is good food for bacteria. How about if you do root canal filling, and the apex is not well condensed and you have some leakage there. The the inflammatory exudate of our body, even if there is no inflammation, interstitial fluid that is there is full of lipid, proteins. That's yummy, yummy, good food for bacteria. So these ecologic factors we have to bear it in our mind, because it is how we try to strive excellence in our treatment. Cofactors, and I alluded to some of them here, especially to mention the microbiological cofactors. pH is very important. I want to ask a question, residents. You have a case, it was irreversible bitis. You open it up, you control the bleeding, maybe a case that has liquefaction necrosis. Anyway, I'm talking about cases where you put your epislocator and it's already out, even you are at the middle, at the beginning of the canal. Why is that? What does that tell you? Well, the canal is dry. What does that tell you? Tell you that the pH is very, very high, as conducting the signal to the end of the root, yet you are at the beginning of the root. So pH is very, very important, and that's not unusual. And usually you have to revert the pH of the canal to be moderate, or to be more basic, in order for the machine to work. So that with the same logic, this is how you're going to suppress bacteria, and that's what calcium hydroxide does. Redox potential, gases in exchange, all of these factors here can or will help and affect the bacteria to domain in one site rather others. That's why, if you ask me now, Dr. Balto, what is the main thing you do when you do root canal? Shaping root canal now is very easy with the heat treated files. And I said, the game now is dependent on irrigation. The art now is how do you irrigate? How and with what? And for how long? And how you mix irrigations together. For me, I think that's what matters. And you think about it, it's with necrotic cases, with the revas cases, even if you are talking about vital pulp therapy. Okay, a big part of the protocol is you control and disinfect the perforation site to the pulp. How do you do that? Without killing the pulp, without hydrating the stuff. So the game now is centered about irrigation and how you control the infection. Okay. Uh, Needless to mention, ladies and gentlemen, that ecological factors will be affected by pulpal and peripheral status and location of the canal. Dr. Hadi, how we're doing with the time? How many minutes do I have?
We've got 20 minutes actually.
20 minutes. Do you want to have some time for questions, or I
Yeah.
finish? Huh?
Yes.
20 minutes for the
After
questions.
20
So I
minutes,
should
we'll have
stop in 10 minutes?
questions. Pardon?
You want to stop in 10 minutes so we can have 10 minutes for questions?
Yes.
All right. All right. All right. So then I want to jump. It's very important. I want to jump to this guy here. All right. All right. Okay. Now I want to finish the ecological factor part, because I want to end up to talk about the focal infection theory, which is very important to me. uh, I usually tell my first 4-year student, the students who are starting root canal here, that I look to the root canal ecological wise and divide them into 3/3: the coronal 1/3, the middle 1/3, and the apical 1/3. Why? Why? Because each of this zone or third has its ecological has its own ambiance. Ambiance is that it all has its own environment. So what is the environment? Well, coronally, it's high oxygen tension, right? It's close to the oral cavity. especially if you have leakage restoration, if the tooth is open and so on. Excuse me. And also in the coronal one third, nutrients are available from the oral cavity. Microorganisms directly exposed to the treatment action. But the more you go down toward the middle third and toward the apical third, they're going to be low oxygen tension. They're going to be reduced amount of nutrients from oral cavity because things are not as close to the source of nutrients as before. And microorganisms are not as readily exposed to treatment like in the coronal one third. Of course, the worst part ecologically is the apical one third. Why? Because it has very low oxygen tension and nutrients are available. Now, you tell me you got far away from the coronal. Why you're telling me there are lots of nutrients? Well, nutrients is a quality nutrients in the apical one third. You know why? Because it comes from the body fluids. Two types of body fluid. Interstitial fluid, if the periapical area is not diseased, or if it's inflammatory exudate, if it's diseased, So these are heavily rich source of nutrients to the bacteria. Microorganism is less affected, and this is where your art, your skill as endodontist comes, that that uh, you are far away. You are not sitting at the corona one third. You are not sitting even in the mouth of the patient. You are sitting outside, and you're trying to reach to that area. That's where the skills come. Of course, people would rather go larger in their preparation. For me, going larger is not art. It's very easy to enlarge the apical one third. What's the art in that? The file does it for you. The art now is to keep that area, as minimum as possible, biologically possible, eradicated from the necrotic tissues and host tissues. Reduce the bacterial colonization as much as you can and seal it well. This is where the art comes. If you when I see now radiographic image of root canal that has lots of gutta percha, then it says, "Ugh, this is ugly. You have destroyed the tooth. You replace the mineralized tissue by plastic. Gutta percha is plastic. And please don't tell me that the art of endodontics is doing that. So, minimum invasive endodontics is a, I'm not saying it's emerging, directive. It's emerged already, and people who are sacrificing tooth structure in the name of treating a disease, they are getting out of the game already. Look it up in the conferences. It's obvious, and the public awareness of this is on the rise. These two pictures, I love them. It's from one of my experiments in mice, not rats. I'm not trying to be another Kakahashi. So the picture on the left is a rat. This is the root of a molar rat that is intact. Intact means control. The one on the right is the a mice that I did pulp exposure for the poor mice. And what can you tell me about the differences between the picture on the right and on the left? On the right, you can see the periapical bone destruction. Imagine this bone was like this before. This is the intact one. You see the periodontal fibers. The periodontal fibers here have disappeared here. And this is what appeared as lamina dura has disappeared, and the periodontal space has widened. This is how it is histologically that you see it in the radiograph. This is the disease. This is the normal. Remember something. Every time you are treating a diseased root canal, you're trying to revert the right one to the left one. See, this is significant. This is significant that you're reverting your all of this, the bone destruction, the fiber that have been taken away in the right image, you bring them back. So when you talk about the ecology, we're talking about look to the diseased area. You see, inside the root canal are the some of the dead tissues and some of the bacteria. So the food and drinks, let me use these words to make it more real. Food and drinks mean nutrients comes from outside, from inflammatory exudate inside the root canal. That's why we don't give antibiotics because they're not going to go inside, except some of the generation antibiotics that has the ability to diffuse inside is very weak evidence on them. Inside the root canal, it's a low redox potential. It's very warm, 37 degrees centigrade. It's dark, it's safe, no noise. The perfect niche for bacterial growth. When bacteria get their nutrients, they start kicking, LPS, bacteria pathology, so on. And you got the cascade of events happening. So just keep these pictures in your mind, ladies and gentlemen, because you will really appreciate the hard task you're doing every time you're treating an apical periodontitis. All right. I want to jump again to this slide. This slide, there's a guy called Sangvest. This study, he wrote it in 2003. Though it's ancient, but you know what was the title of this study that Sangvest wrote? Life as an endodontic pathogen. I love it. So this guy,
Very interesting. Very interesting.
yes, I think Dr. Hadi, maybe he was a friend of Dr. Safranisky. He used to sit
Mm.
like this for hours in front of the patient. And so this guy he lived it in three dimension. He talked about post-treatment periodontitis, and he talks about the location here on the right and microorganism, and he talks about oh my God how life it is. Metapersia is there, coronally there is a crown, and apically there's maybe a breach through metapersia interface, and he thought about it all. Please read. If Dr. Hadi has it or can send it to you, it's a wonderful article where it will maybe take you through the analytical thinking or thinking about things that you don't see by your eyes, but you can imagine it. And imagining it will give you two advantages. Number one, it will bring you closer to knowing what you're doing every day. Second, it's cool. It's very cool, actually, that you can imagine a world that you don't see by your naked eye.
Pretty many.
All right. Okay, doctor, we have how many minutes? We have 5 minutes. Okay,
Yes.
unfortunately, I won't be able to talk about biofilm. Talking to you was very interesting. I want to talk to about something that is very, very important to me, ladies and gentlemen, which is here. Okay, this is endodontic. Uh, okay, why it's not? Wait, wait. Hello, are you with me?
Yes.
Yeah, somehow it's not. Okay, I have a clutch here. Just a second. Here we go. No, it's not. There you are. All right. Okay, this is Endodontics then. I got this movie from a friend from India, honestly. This is how Endodontics was. This was actually, if we go to the fourth site in Boston, they had this old dental chair that, operates by a leg. And this is how the root canal used to be done. And they will fill the root canal with silver points, and they were using formalin, formaldehyde, and it's, it's amazing how things have changed. At that time, there is a guy called Hunter. You see, I'm bringing you lots of names. So we'll talk about Takahashi. Hunter gave a lecture in McGill University in Canada, if I remember well. Yeah. And the title of the lecture was The Role of Sepsis and Antisepsis in Medicine, and the importance of oral sepsis as its chief cause. Hunter started the focal infection theory. Focal infection theory, the gist of it, if you don't know it, I'm sure you know it, that the persons who had systemic diseases, heart disease, liver disease, diabetes, type 2, whatever, the etiology was coming from the oral cavity. Because Hunter speculated that uh, if you have a foci of infection in your body, this can have the ability of leaching out microbes that travels into your body and causes this systemic disease. Believe me, people believed him. At that time, everybody who had root canal treatment problem or periodontitis, they would extract all teeth. It stayed like that for a couple of tens of years. And then when it stopped because not because of evidence. They found that some of the people who had foci of infection did not have systemic disease, and on the other way around, some people who had systemic diseases did not have foci infection in their mouth. And I want to understand why only the mouth. There are other parts of the body that are full of facile infection, like nose, like ear, like urogenital. Why the mouth? I don't understand. Anyway, it kept that way for a while, and until Binder came, and this focal infection theory has been refuted. At the moment, the focal infection has been refuted to be the focal inflammatory theory, not the focal infection theory anymore. The microbiological studies have found there is no way the bacteria that goes outside here can go and live in the valve of the heart. It's different ecology. But what is so strong now, and we are on the hook, ladies and gentlemen, the focal inflammatory theory says or states that if you have a fossil infection in your mesobuccal root of upper first molar, all right, this has the power of leaching out cytokines like interleukin 1, interferon gamma, TNF alpha. These cytokines can travel in the body and cause the effect that can, for example, destroy Langerhans cells in the pancreas and lead to diabetes type 2. Can go to liver, do something to the kidney. This is a very strong theory, and all the data that are accumulating on it says that it can be actually the case. So check it out, the focal inflammatory theory. Okay, last slide here is the clinical management of microbes. Of course, the gist of this lecture is to brainstorm. However, let me review with you. I told you that our best performance now for me as Khaled Balto is the irrigation. Of course, after you do proper diagnosis, access, blah, blah, blah. I mean, what really matters now is the irrigation. And irrigation is a big world. Maybe one day we can have a discussion about it. There's intercanal medication, and calcium dioxide stands up in it. And in spite of all the advancement we had, there are lots of other trials that has tried to substitute calcium hydroxide, but still, until now, it's the golden standard of root canal treatment. Replacement therapy, probiotics, antimicrobial peptide, caries vaccination, lasers, antimicrobial hydrogel, and what else? And all of these are avenues. If you see, the clinical management of microbes is changing tremendously. Laser is very interesting. Its cost is not justifying the cost effectiveness part of it. Probiotics and hydrogels are booming. Here is vaccination. It has been problematic since years. However, maybe we are getting close to having successful CARES vaccination, photodynamic theory. All of these are areas that you have to bear it in mind. For me, my favorite clinical managed microbes intra-radically, radicular, is agitation with passive ultrasonic irrigation with sodium hypochlorite slash other irrigation depending on what you're doing. Revas, of course, you need different irrigation from Revas to post-treatment therapy, post-treatment disease to primary endodontics. All right. With that saying, I think it's about time, right, Dr. Hadi?
Yes, yes, we have.
So this is the last slide here. Ladies and gentlemen, it was a pleasure talking to you, and the floor is yours, Dr. Hadi.
Thank you very much. Thank you very much for your informative lecture. That was very kind of you for joining us today. Of course, we had a lot of online guests to the meeting, and some unsuccessful guests who could not join us because of the technical problems. I don't know why, but I provided everything. But anyhow, we really enjoyed. That was a really good discussion about microbiological aspects of endodontic management of teeth with ulcerative bronchitis and bronchitis. So it was a very good opening for us to collaborate for our future research program. Because as far as I know, we have a large number of ongoing research projects concerning eradication of bacterial species from the canal system, so in different ways, in clinical, preclinical, and experimental models. So we can have good collaboration in this field based on your experience, and I think your experience will be of great value for us. Anyhow, thank you very much for your time and efforts. I think it was a very good opening session, so we can have it continued. And of course, if you agree, we can also have mutual collaborations in this field. Thank you very much for providing us with your contact information so our residents can take picture of your contact information and can collaborate with you in case of any questions. Anyhow, that was very good. I think a very good experience for us today. Uh, have a good time, and I hope to see you in Iran.
Thank you.
So let me open my video. That's a little Indian junction for So I invited all the residents uh, to have a mm, photo with you. This is Simon. These are our residents.
Assalamu alaikum. Hi, ladies and gentlemen.
Some of them can speak Arabic with you.
Oh, really?
Yeah.
حالكم.
السلام عليكم.
عليكم السلام، أهلين، كيف حالك؟
جميل الحمد لله شكرا.
إن شاء الله كانت المحاضرة مفيدة لكم.
شكرا على تجربتكم كلش خريجة كلش مفيدة فهموني. أنا دكتور وسام من العراق.
والله ما سمعتي، ممكن تعيد السؤال لو سمحتي؟
أقول أنا دكتورة وسام من العراق. المحاضرة كلش مفيدة وزينة كانت على تجربتكم
حياك
كلش.
الله. الحمد لله، فرصة سعيدة جدا، أنا مبسوط إنكم استفدتوا.
شكرا.
Dr. Hassan is our international student from Iraq.
Yeah, she told
And
me. Nice.
yeah, the others are Iranian, pure Iranian.
Hi guys.
So let's have a photo here. You guys got that one, you see. Thank you.
Okay, thank you guys.
Thank you. Bye for
Bye.
now.